getting medicines into the brain to treat devastating diseases like Alzheimer's, Parkinson's, Huntington's. It's arguably one of the toughest challenges in all of medicine.
It truly is. The brain is just so well protected, that blood-brain barrier, it acts like this incredibly tight, almost impermeable fortress. Great for keeping bad stuff out, but yeah, a massive hurdle for therapies.
Absolutely. And that formidable barrier, well, that's exactly what we're diving deep into today. Welcome. We've been looking closely at source material focusing on one company, Arrowhead Pharmaceuticals, and their potentially really groundbreaking approach to maybe finally cracking that CNS delivery problem.
Yeah, specifically, we're going to unpack Arrowhead's TRIM platform, focusing on how they might use it to get RNAi therapeutics, these powerful gene-silencing molecules, into the central nervous system, and remarkably, doing it with just a simple subcutaneous injection, a shot right under the skin.
A subcutaneous shot for brain diseases. I mean, that alone sounds like a potential paradigm shift.
It really does.
So our mission in this deep dive is to explore how this tech is designed to work, what the preclinical data, especially those really key non-human primate studies, are telling us, what it could all mean for, the millions affected by these conditions, and really what makes this whole systemic subcutaneous idea potentially a game changer compared to the usual methods.
Okay, right. Let's set the stage a bit. The sources, they start with a quick primer on RNAi itself. You should think of it as this revolutionary way to step in at the genetic level. So instead of trying to target and clean up the misfolded proteins after they're already causing damage.
Which is what many drugs try to do.
Exactly. RNAi uses these small interfering RNAs, SNCAs, to silence the gene responsible before that problematic protein even gets made. It's like intercepting the faulty instruction manual right at the source.
Going upstream, hitting the source code of the disease itself, that's the real promise of RNAi. But, like we said, the massive hurdle is still that blood-brain barrier, the BBB.
Right, and the sources really emphasize just how good the BBB is at its job. It's made of these endothelial cells packed super tightly together, tight junctions, they call them like a sealed wall, stops most molecules just drifting through. And then you've got these active pumps, efflux transporters like P-glycoprotein, sitting there ready to kick out anything foreign that does manage to sneak in.
Optimized for exclusion.
Totally, optimized for exclusion.
And the diseases we're talking about, Alzheimer's, Parkinson's, Huntington's, the global burden is just staggering. The sources mentioned over 50 million patients worldwide, and the economic cost, wow, over $413 billion a year, just in unpaid dementia caregiving costs. It's huge. And these diseases are often driven by these abnormal proteins, building up tau, Huntington, alpha-synuclein.
Which is exactly why RNAi is so attractive here. If you can safely and effectively deliver that SNCA to shut down the genes making those proteins, you could potentially change the whole course of the disease.
Now, it's worth pointing out, as the sources do, that Arrowhead isn't exactly new to this game. They're not new to RNAi or difficult delivery challenges. Their corporate profile shows they're focused on gene silencing across a pretty broad pipeline: cardiometabolic, pulmonary, liver, neuromuscular diseases too.
And that experience is really key, isn't it? Their success delivering RNAi outside the liver, which was kind of the first big target tissue for RNAi, shows they have real expertise in getting these molecules where they need to go. Plus, the sources highlight the significant partnerships they have: Amgen, GSK, Sarepta, Takeda, big names.
Right. These aren't just small deals. They represent, serious validation of their core technology and bring in significant cash, like that $825 million from Sarepta. It suggests they have the technical know-how and the financial stability to tackle something as incredibly tough as CNS delivery.
Definitely gives them credibility.
Okay, so that sets the scene. Let's get into the nuts and bolts. Their core delivery technology platform, which they call TRIM.
Right, the TRIM platform. The sources describe it as highly adaptable, modular. Think of it like a sophisticated toolkit, almost like Lego bricks, designed with interchangeable parts for different delivery jobs.
A customizable delivery vehicle. Okay, what are the key parts then?
Well, you've got targeting ligands. These are like the zip codes, guiding the therapeutic payload to the right cells or tissues. Then there are stabilization chemistries, really important for protecting the fragile RNAi molecule so it doesn't get chewed up in the body too quickly. You've got linker chemistries holding everything together, and specific structures built in to improve the pharmacokinetics and pharmacodynamics, basically, how the drug moves around the body and what effect it has and for how long.
So it's not just sticking a guide on the RNAi, it's a really engineered system.
Exactly. And the sources mentioned they use sophisticated algorithms, computational methods, for picking the best RNA sequence. This helps maximize the silencing effect, boost stability, and crucially, minimize any unwanted off-target effects. That modularity and the computational design, that's a key insight into maybe why they can adapt this platform relatively quickly for different gene targets.
All right, so they've got this versatile TRIM platform, but the next big challenge is getting it across the BBB. The sources talk about different strategies, starting with maybe the more traditional route for CNS delivery, intrathecal administration, or IT.
Yes, their TRIM CNS IT platform. This involves injecting the drug directly into the cerebrospinal fluid, the CSF, which bathes the brain and spinal cord. The whole idea is to bypass the BBB completely by putting the drug straight into the CNS fluid system, letting it spread from there.
Makes sense. Bypass the barrier. But what did the preclinical data, especially in non-human primates, NHPs, which are usually a good indicator for humans, show about this IT approach?
Well, the NHP data was pretty revealing.
Right.
It showed you could get the RNAi into the CNS this way. You saw the highest drug levels in areas close to the injection site, like the spinal cord and the cortex, the outer layers of the brain. And here's the really critical finding from that data.
What was it?
The distribution into the deeper brain regions, places like the brainstem, the pons, medulla, caudate, putamen, and midbrain, it was really limited and highly uneven. The sources actually quantify it. The RNA concentration in these deep areas was up to 30 times, sometimes even 37 times, lower than in the spinal cord or cortex.
30 times lower. Wow. That's not just a little uneven. That's like trying to put out a house fire by only spraying the front door.
That's a perfect analogy. And as you'd expect, the actual gene silencing, the target engagement, it mirrored that uneven spread. You got good knockdown where the drug concentration was high spinal cortex, but hardly any silencing in those deep brain areas that barely got any drug.
So the IT approach, while it gets some drug in, seems inherently flawed for widespread diseases that affect the whole brain, including those critical deep structures. This unevenness, highlighted in the IT data, really sets up the why for Arrowhead, looking for something different.
Exactly. And this is where we get to what looks like the main innovation here, the real focus of the sources. Their TRIM CNS SC BBB platform, the systemic subcutaneous approach.
Right, the one delivered via a simple shot under the skin. How on earth does that navigate from the bloodstream, get across that fortress BBB, and into the actual brain tissue?
Okay, so this platform uses a totally different strategy, non-invasive, and it cleverly hijacks the body's own transport systems. The absolute key player here is the human transferrin receptor 1, or TFR1.
TFR1, I know that one.
Yeah.
Isn't that involved in getting iron across the barrier?
That's exactly right. And here's the clever bit. TFR1 is highly, highly enriched on those endothelial cells that make up the blood-brain barrier. Even better, it's a receptor the cell naturally internalizes and recycles really quickly. This makes it a perfect target for something called receptor-mediated transcytosis.
So Arrowhead designed their drug to basically hitch a ride on TFR1, like a ferry service.
Precisely. The ligand part of their TRIM SC-BBB platform is engineered to bind specifically to TFR1. When it latches on, it essentially tricks the BBB cell into starting transcytosis. That's the process of moving the whole complex across the cell, and endocytosis, bringing it into the cell on the brain side.
It's like having the right key card to get past security because you look like you belong there. The sources call it a biological gatekeeper, or even a Trojan horse.
Yeah, very apt descriptions. And a really important safety point the sources highlight. The way Arrowhead's ligand binds to TFR1 apparently doesn't stop the body's own transferrin, the natural iron carrier, from binding. This is vital. It means their system can ferry the drug across without messing up the essential, normal iron transport the body needs. It's hijacking the pathway without breaking it.
That's clever. Really clever. Avoids potential side effects from disrupting iron transport. So that's the mechanism used TFR1 to get across. But the million-dollar question is, does the data show it actually works? And how well?
Right. That leads us to the preclinical validation, started with mouse models. They used special transgenic mice engineered to have human TFR1 in their brains. When they gave these mice the SC-BBB therapeutic, they saw significantly higher RNA buildup, like over 50 times more compared to normal mice without the human receptor. That really confirms the transport into the brain is happening via human TFR1.
Okay, proof of concept in mice checks the TFR1 box. But we all know CNS delivery is notoriously tricky. What works in mice often doesn't translate. So what about the non-human primate data for this SC-BBB platform? This is where we really see the potential for humans, right?
This is where the SCBBB platform really seems to shine and where the data gets incredibly compelling, according to the sources. The NHP data shows that subcutaneous administration led to thorough and really remarkably even distribution of the CERN, and not just in some areas, but across all the CNS regions they looked at.
All regions. You mean including those deep brain areas, the ones the IT approach basically missed or barely touched?
Exactly. The sources list them specifically. Caudate, putamen, thalamus, hypothalamus, substantia nigra, hippocampus, midbrain, pons, medulla, cerebellum, all those deep structures, often ground zero for diseases like Parkinson's or Huntington's, they were reached effectively.
And the evenness. How did that compare to the, what was it, 30-fold difference we saw with the IT method?
This is probably the most striking contrast in the data. Remember, the IT platform showed that huge 20 to 30-fold difference in drug levels between regions. With the SCBBB platform, the maximum difference they saw across all CNS regions, including deep brain, was only about 2 to 3-fold.
Wow. Okay, that's not just an improvement. It's a fundamental shift.
Yeah.
Going from a 30 times difference down to maybe 2 or 3 times. It sounds like they actually achieved something close to uniform coverage of the entire brain.
That's precisely what the NHP data seems to suggest, a dramatically more uniform delivery profile across the whole CNS landscape.
And did that even distribution of the drug translate into even gene silencing, even knockdown?
Yes. That's the really powerful finding. The NHP studies showed effective target silencing getting more than 50% knockdown of the target mRNA across most regions, and in some areas hitting up to 75–85% knockdown. And crucially, mirroring the drug distribution, the SC injection led to even mRNA reduction across all those brain regions, including the deep ones.
So even delivery leads to even silencing right across the whole brain. That feels like the absolute key breakthrough here, doesn't it?
It certainly seems like it. And the sources add that this knockdown wasn't just deep, it was also durable. With multiple doses in NHPs, they kept knockdown at 50% or more out to 3 months, even in those deep brain areas. This durability is what supports the idea of maybe monthly or even quarterly subcutaneous dosing in humans.
Okay, let's just pause for a second and really connect this back to you, the listener. Why does this NHP data showing this remarkably even distribution and silencing across the entire brain, why does that matter so much?
Well, it matters profoundly because diseases like Alzheimer's, Parkinson's, Huntington's, they aren't confined to one little spot. They are widespread, diffuse conditions affecting many, many interconnected brain regions. So to really have a chance at halting or reversing them, you need a therapy that gets everywhere the disease is, including those deep structures vital for function. Achieving this kind of thorough, even distribution across the entire CNS, minimizing those regional differences, that's the critical differentiator for Arrowhead's subcutaneous approach. It provides a really strong scientific reason to believe it could actually work for these complex, widespread diseases, and all via a non-invasive shot.
It tackles that core limitation of previous delivery methods head on. Okay, so how is Arrowhead actually applying this platform now? They have lead candidates targeting 3 huge neurodegenerative diseases.
Right. First up is ARO-AMPT. This one targets the tau protein, which is heavily implicated in Alzheimer's disease. The NHP data for the specific drug showed deep, durable knockdown of tau mRNA, up to 85% reduction across lots of CNS regions. Spinal cord, cortexes, caudate, putamen, thalamus, hippocampus, pons, medulla. And importantly, it maintained over 50% knockdown in deep brain areas at the 3-month mark.
And based on that pretty solid NHP data, what are they thinking for human dosing?
Well, their modeling suggests that quarterly subcutaneous shots just once every 3 months could maintain a 50–70% knockdown of tau. They also project that after maybe an initial weekly loading phase, monthly boosters could potentially push that knockdown up to around 80%.
Quarterly or monthly shots under the skin for Alzheimer's compared to invasive brain procedures. That's just huge in terms of potential patient convenience and acceptance. And Arrowhead is keeping this one all to themselves.
Yes, the plan is for Arrowhead to retain full global rights to ARO-AMPT. It's a big statement, really. Signals huge confidence in the platform and this specific drug for the enormous Alzheimer's market, 6.9 million patients in the U.S. now, expected to double by 2050. They're clearly aiming to capture the full value here. They're expecting to file the CTA, that's the application to start human trials, by the end of 2025.
Okay, next up, ARO-HTT. targeting the Huntington protein for Huntington's disease.
ARO-HTT goes after both the mutant and the normal Huntington protein. The NHP data showed significant reduction of the protein itself, over 75% knockdown throughout the NHP brain, including key areas like the temporal and frontal cortex and the caudate and putamen, which are really hit hard in Huntington's.
And for this one, they've teamed up with Sarepta Therapeutics.
Correct. Sarepta brings a lot of expertise in rare genetic diseases. Huntington's, while devastating, is rarer than Alzheimer's, about 41,000 patients in the U.S. So this partnership is pretty strategic. It lets Arrowhead share the development costs and risks, leverage Sarepta's specific know-how, and potentially speed up development for ARO-HTT. It also frees up Arrowhead's own resources for those bigger opportunities like Alzheimer's and Parkinson's. And like we mentioned, that partnership brought in significant non-dilutive funding, validating the tech again. The CTA timeline for ARO-HTT is similar, expected by the end of 2025.
And finally, ARO-SNCA, targeting alpha-synuclein for Parkinson's disease.
Right, for ARO-SNCA, the NHP data showed substantial knockdown of the SNCA mRNA, over 70% overall. But interestingly, they saw over 90% deep silencing in really disease-relevant regions like the putamen and the substantia nigra. These are areas critical for motor control and known to be heavily affected in Parkinson's.
Hold on, over 90% knockdown, specifically in the substantia nigra and putamen from a subcutaneous shot. That seems incredibly targeted for a systemic delivery.
It really highlights the platform's potential, doesn't it? Not just to get everywhere evenly, but also to achieve really potent silencing in key areas. Parkinson's is another huge market, 1 million U.S. patients. The CTA package for ARO-SNCA is being put together now, with filing expected in the first quarter of 2026. The sources suggest they're still deciding on the long-term plan here, whether to keep it fully in-house or maybe look for a partner down the line.
So across the board, very promising preclinical efficacy data for these major neurological targets. What about safety? Any red flags in the preclinical work?
The sources mention exploratory toxicology studies in rodents and NHPs. The takeaway seems to be a favorable safety profile so far. A key point they highlight is a significant safety margin, meaning the dose needed for the therapeutic effect was more than 10 times lower than doses that caused any safety concerns. That kind of margin is really important for de-risking as they head towards human trials.
That's encouraging. And the actual drug formulation itself, is it practical for a subcutaneous shot in people?
Yeah, the sources confirm they've developed a formulation that can deliver 150 milligrams of the RNAi drug in a volume of 4 milliliters or less. That's well within the typical range for a subcutaneous injection, making it feasible and potentially patient-friendly.
Okay, let's zoom out a bit and put this in the wider context. The whole market for RNAi therapeutics is, well, exploding, isn't it? Projected to grow from about $5 billion this year to almost $29 billion by 2034.
It's a really rapidly growing field, and neurodegenerative disorders are already a huge chunk of that, over 43% of the market back in 2023. The growth drivers are pretty clear: the rising prevalence of these diseases, big advances in delivery systems like Arrowhead's potentially, the move towards precision medicine, and the success of existing RNAi drugs like Alnylam's Onpattro, which prove the concept works, albeit for a different disease.
So Arrowhead is right in the sweet spot, the biggest segment of a very high growth market.
Absolutely. Now, the field still faces hurdles, right? There's always a need for even better intracellular delivery in some tissues, ongoing concerns about potential off-target effects, high costs, complex regulations. But Arrowhead's engineered TRIM platform seems specifically designed to tackle many of those delivery and specificity issues head-on.
And their specific approach, using TfR1 for transcytosis, that fits within the broader research landscape for getting drugs across the BBB, right?
Yeah, it's a prime example of one of the most promising strategies out there. The sources mention other active areas, nanotechnology, different Trojan horse, or receptor-mediated transport strategies using other receptors, even physical methods like focused ultrasound to temporarily open the barrier. Arrowhead's platform is a very sophisticated take on that TfR1-mediated transport idea.
How do they stack up against competitors, specifically going after neurodegenerative diseases with RNAi? The sources mention names like Alnylam, Atalanta Therapeutics, UMass Chan.
Right, there are definitely other key players. Alnylam, a real pioneer in RNAi, is moving into neurology. Their lead CNS candidate, milosirin for APP, uses intrathecal delivery, though. Atalanta Therapeutics has their own platform, and an HTT program nearing the clinic looks like wholly owned. And academic centers like UMass Chan are doing important work, also often focused on intrathecal delivery.
So what is Arrowhead's core differentiator, according to the sources? What makes them stand out in this competitive landscape?
The sources are pretty clear on this. Arrowhead's distinction comes down to the combination of systemic subcutaneous administration, coupled with that robust preclinical NHP data showing thorough and remarkably even distribution and silencing across all CNS regions, including the deep brain. That minimal 2-3-fold variability with their SE platform is positioned as a major potential advantage over the highly uneven 20-30-fold differences seen with traditional local IT approaches.
So it really circles back to that powerful combination. The sheer convenience of a subcutaneous shot, plus the compelling data suggesting it can achieve unprecedentedly even coverage of the entire brain.
Exactly. For diseases that are spread throughout the brain, getting the drug evenly distributed is absolutely critical. And doing that non-invasively could offer huge benefits for patients and families dealing with these chronic, incredibly challenging conditions.
Okay, so let's try and wrap up the key takeaways from this deep dive. Arrowhead's TRIM CNS SC BBB platform is a novel approach. It uses systemic subcutaneous delivery, cleverly leveraging the TfR1 receptor to get RNAi therapeutics across the blood-brain barrier.
And the preclinical data, especially in non-human primates, looks really compelling. It shows remarkably even and durable drug distribution and gene silencing across the entire CNS, importantly, including those hard-to-reach deep brain structures.
So it really circles back to that powerful combination. The sheer convenience of a subcutaneous shot, plus the compelling data suggesting it can achieve unprecedentedly even coverage of the entire brain.
Yeah, and with promising preclinical efficacy signals and a favorable safety profile reported across programs targeting huge unmet needs like Alzheimer's, Huntington's, and Parkinson's, Arrowhead seems well-positioned to move these potentially transformative, non-invasive therapies into human testing pretty soon.
Ultimately, this isn't just, a complex biotech story about delivery vectors. It's really about potential new hope, a potential fundamental shift in how we might treat these widespread brain diseases, moving away from invasive, regionally limited methods towards potentially more convenient, systemic solutions.
And maybe the really provocative thought to leave listeners with is this. Given this platform's demonstrated ability, at least preclinically, to safely and evenly deliver RNAi throughout the entire CNS with just a simple subcutaneous shot, how might this technology not just change the game for Alzheimer's, Parkinson's, and Huntington's, but potentially unlock therapeutic avenues for a whole host of other widespread, diffuse CNS disorders currently considered untreatable, mainly because getting drugs past that barrier and getting them everywhere has just been too big a hurdle?